So if we go back a second, we'll have a look. There are stop codons and start codons mentioned on this slide. So what are they? So we know that a triplet of bases like this makes up a codon, and each codon will code for a specific amino acid. As it says here, each gene starts with a start codon, and to make a protein, and you must end with a stop codon. So start codon starts the process, stop codon stops the process of the protein being produced. Now, we've already looked at mRNA. Now we're going to look at this thing called tRNA, which is a different type. It's called transfer RNA. And as it says here, it is made of a single chain of nucleotides and is found in the cytoplasm of a cell. As it says, it is folded due to base pairing to form a triplet, something called an anticodon, which we'll discuss, and an attachment site for a specific amino acid. So easier to explain when we're looking at the diagram. So this shape here, you will notice over time that this shape diagram appears quite a lot in past paper questions. And there's two main regions you need to be aware of. One of them is down here, and this is called an anticodon. And you can see that it has three bases, just like a codon. And what the anticodon, what its job is to do is to base pair with a codon. Okay, so that's its main role. And when we discuss translation, that will hopefully make more sense. The other region you have to be aware of is here. Now, you'll notice that this part is higher up than this part. So it's the higher up section of the diagram. And this is the site where the amino acid will bind to. Okay, so this carries the amino acid up here. So sometimes you're asked, whereabouts would the amino acid be? And you would just point to this section here. Okay, right. Ribosomes. We've mentioned that protein synthesis will occur in the ribosome. So translation occurs in the ribosome. That's where the protein is actually produced. As it says here, the ribosome reads the codon and sends a signal for a complementary anticodon with a specific amino acid attached. So if you have a look at this diagram, okay, this large kind of orangey ball section is a ribosome, and you can see that we have our mRNA, which has come from the nucleus, and you can see that we have these structures here. So this is tRNA, this grey thing, okay? Now, this is the anticodon, which is going to base pair with these codons here, and the tRNA carries an amino acid. So when this comes in to the ribosome, it's going to basically bind to the base pairs. Okay, they're going to pair up so that the codons and anticodons base pair. And when that happens, it also brings with it an amino acid. Now, when we look at the stages, you will see what happens to these amino acids. So, here we go. The first step of translation, the ribosome binds to the mRNA at the start codon. So the ribosome in this diagram is this big purple blob rather than an orange one. It binds to the mRNA, which is this long strand here, at the start codon. Next, a complementary anticodon carrying an amino acid attaches. So this is the tRNA. It's got an anticodon at the bottom, and it's carrying this blue thing, which is an amino acid. It binds to its complementary codon. You can see that they're complementary base pairs. Then the next mRNA bonds with complementary anticodon. Okay, so another tRNA with the correct amino acid, this one here, will come and it will bind here. Okay, because it has a complementary base pair. So the anticodon codes the codon. Okay, but it also brings this amino acid. So now you're going to have this amino acid and this amino acid beside each other. And what happens when you have two amino acids beside each other? They form a bond. A peptide bond forms between them and joins them together. Okay,